Let's talk about viruses. Viruses, by definition, are obligate intracellular parasites. So let's talk about what this means word by word. Obligate meaning by necessity, intracellular means inside of a cell, and parasite means dependent on that host cell that the virus is inside. And what makes a virus dependent, what makes it a parasite, is that the virus has no organelles or nuclei of its own. So it's not able to make its own ATP and it's not able to replicate its own genome. It's completely dependent on the organelles and the functions of the host cell to be able to maintain its life. And viruses are very small. On average, they're about 20 to 300 nanometers. This is teeny tiny. So small, it's about 100 times smaller than bacteria and 1,000 times smaller than eukaryotic cells. So this is so small you can't see it with a regular microscope. You have to use electron microscopy to be able to observe most viruses. And all viruses have two things. They all have a nucleic acid genome, and they all have a protein capsid. Some viruses have a third thing, and this is an envelope. Now going back to the genome can be one of two things. It can be either DNA or RNA, and that's the nucleic acids. So the genome is made of nucleic acid, which can be either DNA or RNA. Now, all DNA viruses are going to be double-stranded DNA viruses. Now, remember this because the D in DNA goes with the D in double-stranded. The only exception to this rule is parvovirus. So all DNA viruses are double-stranded except parvovirus, which is single-stranded. And if all DNA viruses are double-stranded, this leaves all RNA viruses to be single-stranded viruses. The only exception to this rule is reovirus. So all DNA viruses are double-stranded, D goes with D. The only exception to this is parvovirus. And all RNA viruses are single-stranded, and the only exception to this is reovirus. Moving on to protein capsids, the function of the protein capsid is to surround and protect that nucleic acid genome. And it's going to be made up of capsomeres. So capsomeres are the subunit of the capsid, and they're going to come together to form the viral geometrical symmetry. And this can be one of two things. So the viral geometrical symmetry is just the shape that the virus is going to take on. And the two shapes that it can take on is first icosahedral or second helical. So it's going to be one of these two shapes. Icosahedral is a spherical shape. If you break it down, it actually has 20 triangular faces and 12 vertices, but generally speaking, it's a spherical shape. And helical, just like it sounds, is going to be a rod-like shape, so it's a hollow coil. So the virus can have two shapes. It can either be icosahedral or it can be helical. So here I've drawn out the nucleic acid genome, this pink structure. And here you can see a capsomere. Whenever you have multiple capsomeres come together, this is going to form the capsid, which is going to, like we said, surround and protect the nucleic acid. And this capsid looks like it's a helical capsid, so this virus is going to be a helical virus. Now, capsomeres have two essential functions. The first, like we've mentioned, is to protect that genome, and the second is to mediate the viral attachment to the host cell. So the host cell is going to have receptors on its surface that the capsomeres of the viral capsid are going to attach to. And this is what's going to allow that virus to be taken up into the host cell. So this is a very important function of the capsomere. The third thing that some viruses have is an envelope. And the envelope is a lipoprotein membrane that's going to surround the capsid. And that lipoprotein membrane, that phrase should be setting off some bells because this is what the plasma membrane of cells are made of. And this makes sense because the lipoprotein membrane actually comes from the host cell membrane. And you can see the blue envelope enclosing the capsid here. So as the virus is taken up by the host cell, it's going to take some of that host cell membrane to become the viral envelope. So that's why it's the same component. That lipoprotein is going to make up the envelope, and it's going to be the same lipoprotein that makes up that host cell membrane. So like we've talked, we can have viruses that are enveloped, or we can have viruses that are non-enveloped. And these non-enveloped viruses can go by two other names. Non-enveloped viruses can be called nucleocapsids, which also makes sense because nucleocapsid is simply describing what the virus is made of. It's made of the nucleic acid genome, and it's made of the capsid. Or non-enveloped viruses can simply be called naked viruses. So you can have an enveloped virus, or you can have non-enveloped virus, also called nucleocapsid or naked virus. So we've talked about all the different structures of the virus, and these are all the main things that you need to understand for the MCAT.